Hi, my name is Dr. Kay Hodgins. I'm a senior lecturer in ecological genomics. I have several PhDs and postdocs in my lab. And probably many of us are interested in how plants can rapidly adapt to climate variation and the genes involved in that. And this is important, right? This rapid adaptation to climate can be important. For example, with invasive species, we often see them expanding their range really rapidly across broad climatic conditions. And this could potentially involve rapid adaptation to climate, genetic adaptation. Alternatively, we might want to help our native species adapt rapidly to changing climates. So I have many honors projects available. For example, I have a large data set, genomic data set, to identify if species can actually borrow genes from one another via hybridization to aid invasion and adaptation. But I'm quite excited about this project that I'm going to tell you about next, because I think it's quite unique and people haven't really done this before. And this is to identify genes important for contemporary climate adaptation in an invasive species. And it's this invasive species here, common ragweed. So in annual plants, we often see this pattern where we have plants from short growing seasons. They tend to flower early at a small size, versus plants found in long growing seasons, they'll delay their flowering until they can get big and have more photosynthetic resources to invest in reproduction, and they'll flower later, right? So we see this sort of life history trade-off where there's two strategies. If you're in a short growing season, you flower early at a small size. If you're in a long growing season, you flower later at a large size. And so these are different optimal strategies we expect to see, and this should be related to latitude, right? Because latitude or high latitude locations, they tend to have very short growing seasons. So we expect something like this, where we have plants flowering early at a small size. And when we have low latitude locations, the growing season tends to be much longer, and we should expect to see this, where plants will delay their flowering and flower at a larger size. And this pattern reflects genetic adaptation. It has evolved in response to climate variation in many, many different plant species. And it has also evolved in this plant species, common ragweed, and again, it's an invasive species, it's native to North America. In the native North American range, we see this pattern again in areas like Florida, which have a long growing season. We see that they flower much later at a large size, whereas areas like Northern Ontario and Quebec, the plants flower when they're really, really tiny like this at a small size. What's really cool about this story is that not only do we see it in the native North American range, but we see this pattern of evolving in Australia and Europe, where it has been introduced. And so over the past 100 years or so, populations in the introduced ranges have adapted to their local climate. Where, say, plants in northern Germany, they'll also flower when they're very small after only a couple of weeks, whereas plants, say, in Queensland, will delay their flowering for many, many, many days and flower when they're taller than me. And this is really cool because we see adaptation to climate occurring so incredibly rapidly. And not only do we see it at the phenotypic level, when we measure plants grown in a common garden in the greenhouse, but also when we look at their genomes. So what you can do is you can identify genetic variants all across the genome in plants from across the latitudinal gradient. Right? And then we can see if there's correlations between the alleles at those different sites and the environmental variable. And this is an indicator that these regions of the genome are actually contributing to climate adaptation. And when my graduate student did that, so she's Lotta Van Bohemen did this greenhouse work and then just did the genomic work, we find that many of the same candidate loci, genes likely important for climate adaptation in North America, are also candidate loci, show these same kind of patterns in Europe and Australia. So there's been rapid adaptation. We can identify phenotypic level right down to the genetic level. So the individual regions of the genome are important for that. So this is really cool. We see adaptation happening really quickly, but I want to see if we can observe adaptation happening over even shorter timescales. And this is in response to contemporary climate adaptation or contemporary climate change. So what we've done is identify genetic variants across all the genome in this species. So millions of sites across the genome, and we sequenced hundreds of specimens from across North America. So we went out and we collected populations from Quebec all the way down to Florida, right? And we can use those data in order to identify very precisely regions of the genome are likely involved in climate adaptation using approaches like this. But what's really cool about this data set is that we've also done the same thing using these herbarium specimens. So these are essentially museum specimens from 1900 to 1950. And we've collected them and sequenced the DNA from them. And this is hundreds of samples from the similar locations as contemporary ones. And what we want to see is if we have these regions that are likely important for climate adaptation in contemporary samples, can we see expected shifts in allele frequency representing adaptation to contemporary climate change? So we already know that in these regions, the growing season is getting longer, has gotten longer because of contemporary climate change. And we can look at contemporary records of pollen production in these species and see that actually this species is flowering earlier already because of climate change. And so can we look at the genetic level and see shifts in our candidate loci that would be consistent with contemporary climate change adaptation? So I think this project is super cool, and I'd be keen on an interested graduate student or honor student working on it. So thanks.